Why are dogs so different in size? This is a legacy from the ancient wolves. From tiny chihuahuas to huge Great Danes. Humans' best friends vary in size more than any other mammal species on the planet. Now the source of the mutation that may be responsible for this variation has been identified. It comes from ancient wolves. The mutation is near a gene called IGF-1, which was identified by scientists 15 years ago as playing an important role in the size variation in domestic dogs. It was the first of about 20 such genes discovered to date. But efforts to pinpoint the specific gene variant responsible for the mutations have been unsuccessful. New discoveries in this area are brought by an article published in Current Biology. IGF-1 has been a thorn in our side, says Elaine Ostrander, a geneticist at the U.S. National Human Genome Research Institute in Bethesda, Maryland, who led the 2007 study that first identified IGF-1's role and is lead author of the new paper. Ancient dogs are domesticated wolves that have evolved over the past 30,000 years. Years differed slightly in size. The current extreme differences. The largest breeds are up to 40 times larger than the smallest, have emerged over the last 200 years and have been created by humans. Ostrander and her colleagues, including geneticist Jocelyn Plassais of the University of Rennes in France, analyzed the genomes of more than 1,400 canids, including ancient dogs, wolves, coyotes and 230 modern dog breeds. Researchers compared variation in the region around the IGF-1 gene with body size in dogs and wild canids. It turned out that one variant is unique. It is located in a section of DNA that encodes a molecule involved in controlling the level of the protein IGF-1, a powerful growth hormone. Scientists have identified two versions, or alleles, of this variant. Across all breeds, dogs with two copies of one version tended to weigh less than 15 kilograms, while two copies of the other version were more common in dogs weighing more than 25 kilograms. The dogs with one copy of each variant tended to be medium-sized, Ostrander says. Dogs with two copies of the large body also had higher levels of IGF-1 protein in their blood, compared to dogs with two copies of the small allele, he adds. When researchers looked at the genomes of other canids, they found a similar relationship. It's not just a story about dogs. We found the same in wolves, foxes, coyotes and all other canids, says Ostrander. Scientists believe that the version of the gene associated with dogs' small bodies is evolutionarily much older than the one responsible for height. Coyotes, jackals, foxes and most other canids had two copies of the small allele, suggesting that this version was present in a common ancestor of these animals. It is unclear when the version of the gene responsible for large body size evolved. Scientists have discovered that the ancient wolf, which lived in Siberia about 53,000 years ago, he carried one copy of this version. Other ancient wolves and modern gray wolves each have two copies, suggesting that the large-bodied version of the gene may have been an evolutionary advantage for them. Until now, Scientists believed that small body size was probably related to relatively new genetic changes, potentially unique to domestic dogs, says Robert Wayne, an evolutionary biologist at the University of California, Los Angeles. This discovery refutes the previous theory. And that's the best thing about it, adds the researcher. The new discovery suggests that dogs were domesticated from smaller-bodied wolves, unlike today's gray wolf populations, said Eleanor Carlson, a geneticist at the University of Massachusetts Chan Medical School in Worcester. We don't even know what the wolves looked like when they turned into dogs, he adds. Scientists emphasize that research on the size of dogs is far from finished. Plassis wants to see how different variants affect IGF-1 protein levels. However, alleles themselves are not the only factors that determine a dog's size. The IGF-1 gene itself is responsible for approximately 15%. Differences between races. Artificial intelligence has solved one of the most difficult problems in biology. A long-standing and extremely complex scientific problem regarding the structure and behavior of proteins has been solved by the new AlphaFold artificial intelligence system. DeepMind scientists have demonstrated that their artificial intelligence can predict what three-dimensional shape proteins will form from amino acid sequences. Predicting the three-dimensional shape of a protein has puzzled scientists for half a century. The ability to accurately predict protein structures based on their amino acid sequences would be a huge boon for life sciences and medicine. This would greatly accelerate efforts to understand the building blocks of cells and enable faster development of new drugs. The problem was solved by artificial intelligence developed by the DeepMind team. It is a company related to Google and has considerable achievements in creating advanced algorithms. A few years ago, their AlphaGo program defeated the Go champion several times. Another of their artificial intelligences, known as AlphaStar, turned out to be better than 99.8%. Players in the real-time strategy game StarCraft 2. However, the achievement of their new artificial intelligence, AlphaFold, beats the good results in games. AlphaFold has made a giant step in solving one of the greatest challenges in biology, determining the three-dimensional shape of a protein based on its amino acid sequence. Artificial intelligence developed by a team of DeepMind researchers beat out about 100 other teams in the biennial CASP, Critical Assessment of Structure Prediction, competition to develop systems capable of predicting the three-dimensional shape of proteins. In the competition, teams of researchers receive amino acid sequences for about 100 proteins and, based on them, have to develop their structure. The results obtained by the teams are compared with the results of laboratory work. Experts are impressed with AlphaFold's work. They indicate that this will have far-reaching effects, including a radical acceleration of the creation of new drugs. What the DeepMind team has achieved is fantastic and will change the future of structural biology and protein research, says Janet Thornton. Director Emeritus of the European Bioinformatics Institute. This is a 50-year-old problem. I was beginning to think it wouldn't be solved in my lifetime. Adds John Moult of the University of Maryland at Shady Grove and co-founder of CASP. AlphaFold took part in CASP for the first time in 2018. She took first place in the final table of the competition. But the algorithms of London's DeepMind stunned everyone this year. Even the competition pointed out that AlphaFold worked so surprisingly well that it could herald a revolution in biology. This is a game changer. This will change medicine. It will change research. It will change bioengineering. This will change everything, emphasizes André Lupas, a biologist from the Institute of Developmental Biology, Max Planck in Tübingen, Germany, who assessed the performance of various teams at CASP. For about 50 years, scientists have been trying to predict how proteins achieve their three-dimensional structure. It sounds simple, but it's not an easy problem to solve. The astronomical number of potential configurations is so astonishingly large that scientists working on the problem have indicated that exploring all possible molecular arrangements would take longer than the age of the universe. Proteins are the building blocks of life, responsible for most of what happens inside cells. The human body uses tens of thousands of different proteins, each of which consists of several dozen to even many hundreds of amino acids. The order in which these amino acids are arranged in the three-dimensional structure dictates their function. In other words, shape determines function. And proteins tend to take shape without anyone's help, guided only by the laws of physics. Knowing these shapes helps scientists develop drugs that can settle into the pockets and crevices of proteins. The ability to synthesize new proteins with the desired structure could, for example, accelerate the development of enzymes that produce biofuels and decompose plastic waste. For decades, scientists have deciphered the three-dimensional structures of proteins using experimental techniques such as X-ray crystallography or cryo-electron microscopy. But such methods take months or even years and do not always work. Thanks to them, structures were developed and functions were understood for only about 170,000. Of over 200 million proteins discovered in various forms of life. The first complete protein structures were determined in the 1950s using X-ray crystallography. It was thanks to this technique that the lion's share of protein structures were determined. However, over the past decade, cryo-electron microscopy has become a favorite tool in many structural biology laboratories. In the 1960s, scientists realized that if they could enumerate all the individual interactions within a protein sequence, they could predict its three-dimensional shape. With hundreds of amino acids in a protein and the many ways each pair of amino acids interacts, the number of possible structures per sequence is enormous. Of course, researchers turned to computers. but early attempts to use them to predict protein structures in the 1980s and 1990s yielded poor results. In 1994, Molt and his colleagues launched CASP as part of the competition. Although Molt prefers to call CASP an experiment, participants are tasked with determining the correct structure of approximately 100 as yet unknown proteins based on amino acid sequences. In CASP, results are given on a scale from 0 to 100, and a result above 90 is treated on an equal footing with experimental methods. By 2016, competing groups achieved scores around 40 points for more complex proteins, mainly by drawing conclusions from known protein structures. When DeepMind first entered the competition in 2018, its AlphaFold algorithm was based on a similar benchmarking strategy. But artificial intelligence also used another approach called deep learning. She learned from huge data sets, in this case, the sequences and structures of already known proteins. She also learned to spot patterns. In 2108, DeepMind won easily, beating the competition by an average of 15% on each structure and scoring 60 points for the most complex proteins. However, these forecasts were not very accurate. To improve AlphaFold's predictions, John Jumper, who leads artificial intelligence development at DeepMind, combined deep learning with an algorithm that mimics the way humans put together a puzzle. First combining pieces into small groups, in this case clusters of amino acids, and then looking for ways to combine the smaller groups into greater whole. AlphaFold, with new assumptions, underwent training on a databank containing approximately 170,000 protein structures. The new tactics and training applied to AlphaFold by DeepMind scientists brought results. In this year's CASP, AlphaFold achieved an average score of 92.4. Although the algorithm performs sometimes better and sometimes worse. About two-thirds of the predictions did not differ from the results obtained using time-consuming laboratory methods. This incredible result drew the attention of the competition organizers to a possible fraud attempt by DeepMind. So Lupus presented AlphaFold with a special challenge. A membrane protein from an ancient group of microbes. Lupus' team tried every trick in the book to figure out the structure of this protein. Their research lasted 10 years and nothing. But AlphaFold had no problems with the task. It returned a detailed image of a three-part protein with two long, spiral arms in the center in about half an hour. As a condition of joining CASP, DeepMind, like all other teams, agreed to reveal enough detail about their method so that other groups could replicate it. This offers enormous opportunities for researchers who will be able to use accurate structure predictions to make sense of opaque X-ray data. It could also enable drug designers to quickly determine the structure of each protein in new and dangerous pathogens such as SARS-CoV-2, which could prove to be a key step in the search for a molecule that will block the coronavirus. AlphaFold's achievements are spectacular, but the artificial intelligence probably has shortcomings. The competition noted mistakes in determining the structure of amino acids, which distort each other's positions during assembly. Jumper wants to train AlphaFold to solve structures like these. as well as protein complexes that work together to perform key functions in the cell. It's not over yet. This is the beginning of many new things, says Thornton. Why is yawning so contagious? Scientists are closer to solving the mystery. The mere sight of a person yawning causes many of us to start yawning too. And we are not alone. Other animals, such as chimpanzees and lions, can also become infected with it. Although scientists think they understand the physiological purpose of yawning, it is still unclear why it is so contagious. There's nothing strong about yawning. Just thinking about it makes us want to open our mouths and yawn for some reason. Andrew Gallup, an evolutionary biologist at the State University of New York Polytechnic Institute in the US, has spent years trying to find out why we yawn. In an article published in Animal Behavior, he reports some evidence of how contagious yawning may have evolved to keep us safe. The scientist analyzed previous research and combined their results into one model. According to Gallup, yawning may be animals' way of synchronizing their behavior and promoting collective alertness. Like another strange reflex, the hiccup, yawning doesn't seem to have an obvious purpose. It was once thought that yawning was a way to replenish oxygen or remove carbon dioxide. Currently, it appears to be more about regulating blood temperature to cool the brain. This method of thermoregulation must be very important. Virtually all organisms with a backbone yawn, from mice, through monkeys, fish, to flamingos. Yawning is therefore a function that probably evolved in our common ancestors a very long time ago. Inducing successive yawns between individuals is a behavior so contagious that it can cross species barriers. This suggests a benefit to the brains of different species that cool together. Yawning usually happens when we change from one state of activity to another, whether lying down to rest or waking up after a long sleep. We also yawn in anticipation of change, arousing ourselves or maintaining arousal when the environment is unlikely to be stimulating. A quick cooling of the brain caused by an influx of fresh air can be a great way to give yourself a short jolt in preparation for a possible task, without switching the brain into fight or flight mode. By sharing a yawn, a group of brains can take responsibility, increasing alertness when one or more group members show signs of fatigue. In an interview with Science magazine, Gallup recalls an experiment he conducted last year. We showed people boards with pictures. Some of them contained stimuli evoking a state of danger, snakes, while others contained neutral images, frogs. We measured how quickly subjects could pick out these images after watching videos of people yawning or moving their mouths in other ways. Gallup explains, after watching other people yawn, the ability to recognize and detect snakes, i.e. threatening stimuli, quickly improved. After observing yawning, The detection of frogs did not change, says the scientist. Uncovering the secrets of yawning may tell us a little more about the subtle forms of communication within and between social species, including our own. A system that delivers sound directly to your head. Thanks to the technology presented by the Israeli startup, you can send sound directly to your ears, without the need to use headphones or earbuds. The SoundBeamer 1.0 device, as its creators put it, creates an acoustic bubble around the listener's ears and no one else can hear any sounds. The system developed by the startup Noveto Systems uses an array of sensors to locate the position of the ears. Finding the target area allows you to transmit sounds that no one else can hear. Interestingly, the device tracks the position of your head while listening to make changes to the position of your ears, allowing you to listen to music while moving. However, you must remain within range of the device's sensors. SoundBeamer has a series of sensors that locate and track the position of the user's ear. Once the ear is located, the equipment sends sound using an ultrasonic wave emitter, creating sound pockets surrounding the ear. These waves are generated by digital signal processing algorithms. The sound obtained in this way can be heard in stereo or as 3D surround sound. The device also allows sound to follow the listener as he moves his head. Associated Press journalists, to whom the company provided the device for testing, described it as straight from a science fiction movie. The SoundBeamer device sends three-dimensional sound that gives the impression that it is in the ears, in front of them and behind them. At the same time, they described their impressions. Journalists watching nature films using SoundBeamer also added that they actually felt as if they were part of the scene they were watching. SoundBeamer has many practical applications, from allowing office workers to listen to music or make conference calls to comfortably playing video games in a room where others are watching a movie. Noveto Systems representatives claim that SoundBeamer is the first consumer product with this sound transmission technology. They also claim that this technology will change the way we perceive sound. However, there are also some limitations. The device, or rather its sensors, must have a clear path to the listener's head. Nothing can stand in their way, otherwise ear position monitoring may fail.